So we're now recording, which means I will post the video after class. So if you need to make reference to the lecture slides or what was actually said during class, you can then go back through and look through those videos. Okay? It'll get posted to YouTube. I don't know exactly where it's going to get posted or how it'll get posted, but I'll try and post an announcement about that so you're at least aware of where you can find it. Okay? So that'll go on to Blackboard, some information about it. Okay? Kind of make sense? Okay, so back with intermolecular forces. So this is what we spent a whole bunch of time talking about last week. And I said we're going to continue to talk about it. So I drew everything up. Now we've got a nice figure to look at. Okay? The goal of this figure is to show the relationship between bonds and forces. They are distinct and different things, regardless of the names that we give them. For instance, hydrogen bonding is a force, okay, not a bond. Okay, so make sure you keep that distinction separate. Where it becomes important or relevant is if, say, you look at a structure, okay, you've got an idea of what bonds are in there. So if you said had a carbon-hydrogen bond, you could classify that according to ionic, polar, covalent, or covalent. And if we have a carbon-hydrogen bond, what type of bond would that be? Where does that typically get classified? It's typically classified as covalent. And that was not a circle. So let's try that again. Okay, I still got to get used to playing around with this. So we've got a covalent bond. Okay, and I picked a nice color. It's a purple covalent bond. Okay, what does that mean about the force that that's going to exert out into the environment? It's going to be a London dispersion force, which is directly across from it. If we picked a purple covalent bond, we'll end up with purple London dispersion forces. Okay? The nice thing about this thing as well is we've got a sequence of strength running up the side. Okay? So as we go up and around, we're increasing the strength of the interaction. Well, if the interaction gets stronger, what happens to the polarity of the molecule, or the polarity of that bond, I should say? It's also going to become more polar. Okay? We can apply that knowledge to a variety of different techniques. For instance, with TLC, we looked at RF values. Okay? Well, what is a good question we could ask for TLC? We could say the lowest, let's see if I can zoom in here a little bit better for me, the lowest RF value. And we're going to compare the structures water versus methane. We want to know which one's going to have the lowest RF. Well, what could we do? Lowest means what? Okay, it means more polar. Right? Hey, that's kind of written on there. More polar. Okay? So if it's more polar, we're going to expect a stronger intermolecular force. Okay? So if we've got a strong force, what type of bond would we be looking for? Something that's ionic. So we could real quickly look at our two compounds, water and CH4. Do we have ionic bonding or ionic forces? No, why not? Do you see ions? No. Do you see a metal and a nonmetal? No. What does that mean? Not ionic. What's the next bond that we would need to look for? The bond type. We'd have to look for a polar covalent bond. Okay? So we can now go back to our structures and find something that has a polar covalent bond. You go through and take a look at the bond structure for water. You've got a hydrogen-oxygen. What type of bond is that?
Polar covalent.
Polar covalent. Okay? We can go to CH4. What type of bond is between a carbon and a hydrogen? Covalent. Covalent, non-polar. We needed a polar covalent bond. Water's going to have the lowest RF. Okay? So an application of those forces will determine what happens with our TLC. Okay, there are other important physical properties that come out of this. Melting point, boiling point. So determining when a substance is actually going to melt or turn into a liquid. All of that's due to the intermolecular forces holding the individual molecules together. Okay? We could look at solubility, which is a large portion of what's happening with today's lab. That's why it's in bold. And we could also look at reactivity. So the second half of this semester is looking at individual chemical reactions. If you understand the bonds and forces and the types of interactions that can come out of these, you can start to predict the chemistry without knowing anything else. Okay? Just based on the intermolecular forces generated between these bonds and forces. Okay? As far as the lab goes, we've got several techniques that are going to come out of this. TLC, which we've already looked at. This week, or starting next Monday, you're going to look at extraction and column chromatography. Both of those rely on intermolecular forces. The week after that, you're going to look at recrystallization, also dependent on intermolecular forces. After that, you're going to look at synthesis, also dependent on intermolecular forces. Okay? So every single one of these things has some role to be played back with these intermolecular forces and the bonds. Okay? Are you tired of me saying intermolecular forces yet? Okay, that means we're going to talk about it some more. Okay, because you clearly haven't listened enough. Okay, so we'll go ahead and move on. We'll take a look at today's or next week's lab. What we're going to do is look at the composition of spinach. Okay, everybody says, oh, organic chemistry has no application whatsoever to the real world. I'd never want to use it. Okay, a lot of drugs, particularly cancer drugs, come from natural sources. Okay. Some of the more famous ones come from actual plants. Okay? That plant produces a very small amount of that drug, and if we want to get access to that drug, we're going to have to somehow crush up and remove the drug from the plant. Okay? We can't just politely go up and say, give me the drug. Okay? It doesn't quite work like that. So we're going to have to somehow pull that chemical out of that structure or out of that plant. Okay? So what we're going to have to do is look at what makes up that spinach leaf. Okay, what pieces go into it? In this case, I happen to pick 4 representative structures, okay, of things that could potentially be found within spinach. Okay? We're trying to get those separated out and come up with some way to purify it. So what's the very first thing that we could do? Rhymes with horses or horses. We could take a look at the intermolecular forces in each of these structures. Which of those might be the most polar? It's a hint. Look towards the bottom of the screen. The bottom one, good call. Okay, that one is the most polar. Why is that one the most polar? We see an ion there, negative charge, right? Right in that lower corner. So that would mean ionic forces. We're looking at something that's very polar. But wait, you say. But wait, you say. What do we see up here at the top? Another ion. Why is that one not as polar as, say, the one down below? The ion is embedded in the middle of the structure. That intermolecular force can't reach out and touch other things because it's trapped within inside the structure. Okay? Those of future budding biologists